Good morning. We warmly welcome you all to the second day of International Virtual Conferences on Chemical Sciences 2022. I'd like to call Dr. M. Rajeshwari, Assistant Professor, Department of Chemistry, to introduce our speaker, Dr. Kamraj Sattu.
Thank you, ma'am. Good morning to one and all. It is indeed a great pleasure and honor to introduce our invited speaker of this session, Dr. Kamraj Sattu. We are honored and would like to express our gratitude for accepting our invitation to deliver the lecture for this session. Dr. Kamraj Sattu is working as an assistant professor, Department of Biotechnology in Periyar University at Dharmaburi. Awards and titles conferred by Dr. Kamraj Sattu. He has received Young Scientist Award in the year 2018 in International Conference on Emerging Areas. Postdoctoral Research Fellowship in the year 2010. Senior Research Fellowship from Indian Council of Medical Research in 2008. Best Poster Award in International Conference on Advances in Free Radical Research, Natural Products, Antioxidant and Radio Product in 2009. Postdoctoral Research Fellow from 2013 to 2015 at Umeå University, Department of Medical Biochemistry and Biophysics at Sweden. He presented many papers in scientific meetings and conferences. He has published many papers in both national and international level. Technical expertise in cell culture and transfection, expression and purification of recombinant proteins in bacteria and baculovirus expression system, plasmid DNA preparation and transfection. We once again welcome you, sir. We would like to take over the session on polyphenols and their effects on enzyme inhibition, a solution for serious disease.
Yeah, thank you so much, ma'am, for a wonderful introduction about me. Can you see my slides?
Yes, sir. Yes, sir.
Yeah. Shall I start?
Yes, sir. You can start the slideshow and you can start. Yes, sir.
Yeah. Good morning, everybody. And I would like to say thanks to the AYURVEDIC College for giving me this wonderful opportunity to present the present my seminars. So today I'm going to talk about the polyphenols and their effect on enzyme inhibitions. So enzyme inhibitions, so enzymes are playing major role in various serious diseases like cancers or inflammations and cardio productions, so many. So today we are going to see about what is polyphenols. Before that, before this knowing about the polyphenols, we are using from the thousands of years these polyphenols like herbals. So we call it traditional medicines, right? So the traditional medicines are more effective and less toxic, even sometimes no toxic, these molecules we used in the traditional systems. Those molecules could cure and control the different ailments. So these chemicals, these molecules are present in the plants, formed in the basis of sophisticated traditional medicine systems that have been in existence for thousands of years. According to the World Health Organizations, they said still 80% of world population still use traditional medicines, either singularly or in combinations with some metals or any other molecules to treat and diagnose or prevent the illness. People who use traditional remedies may not understand what is inside the medicines and what is the mode of mechanisms these medicines would treat the diseases. but they don't know, but still they are believe and used these medicines. Mostly in Asian countries like China, India, and they are right system by the government of India by Ayush, right? This name of Siddha, it owes its origin to medicinal idea and practice of classes of Tamil sagas like called Siddhas. Siddha medicine is defined as a constitute of death or ensure prevention against the mortality. What is the materials used in Siddha? We called it herbal, right? We call it Mooligai Tavaram. In not only the herbals, they also include inorganic substance called it Tadu and animal product called Jiva Vagappu. The pure herbs are mixed herbs with metals, minerals, and animal products used to treat different diseases. This is like 20 ancient siddhas. We all know that. So not only siddha, we have to use other traditional medicines like Ayurveda, Unani, homeopathy. These are all the alternative medicine systems, alternative for the allopathy. But right now we are using the allopathy medicine. Simply we call it English medicines, right? So the Ayurvedic medicines, Ayurveda is the science of life. Ayurveda means science of life. These Ayurveda working based on this Panchapota. It perhaps the most ancient of all traditional medicines, probably the older Chinese traditional medicines. This is like disease or disorder are thought to be caused by physiological imbalance. Either this Panchapota are imbalancing the disease, what they believe according to the of Ayurveda. It could be from this imbalancing of this this disease. It could be from the poor food habit and also environmental changes or shock to the human systems. How they are diagnosis in the Ayurveda means they are using pulse reading, physical examinations, and appraisals of symptoms. Allopathy medicines, we know that it is English medicines, right? This is also based on the germ therapy of disease and the study of anatomy and physiology. They are also monitoring health and normality are dependent reference of certain physical and biochemical parameters. What we used in the laboratory, what the testers are taken in the laboratory, right? So the tester, based on the laboratory test, they can go for the treatment. This is like a weight, height, blood pressures, and the heart rate, etc. So what is medicinal plant? So we are talking about the polyphenols, right? So what is the relationship between medicinal plant and the polyphenols? Means polyphenols are source of medicinal plants. So that's why we want to know what is medicinal plants. So medicinal plants having the pharmacologically active compound. These compound exploited in traditional medicines for treatment of various ailments. This active compound can able to provide offer an alternative synthetic drug. But right now, what we are taking is synthetic drug, right? In the allopathic medicines, these medicinal plants are alternative to the current synthetic drug or synthetic drug substance in preventing and treating some chronic or mild diseases. The quality of herbs depend on the species variations. What is species variations? The species variations, for example, we are using chili, right? Red chili we are eating. Some chilies are more alkaline, some chilies are less. So these, for example, chili containing capsaicin. Capsaicin is one of the alkalides. These capsaicins, it's used by the treatment of some diseases. So the chili, the capsaicin is based on the alkaline. Some chilies are more alkaline, some are less alkaline. That's why the species variations and also environmental conditions and the time of harvesting. Time of harvesting is one examples we know. The tomato, we are using tomato, right? Tomato having the lycopene. The lycopene only developed after ripening of tomato. So the time of harvesting is very important. And also the storage and processing also determine the quality of herbs what we choose. So India have a rich flora that is widely distributed throughout the country. This map showing how this tropical and subtropical, moisture and dry broadfield forest, especially in Himalayas and also in Western Ghats in South India and also Northeast, having the good source of floral distributions. So what is phytochemicals? Can you hear my voice? Anyone? Can you hear?
Yes, sir. Yes, sir. It is clear. Sir, your voice is clear, sir.
Okay. Thank you so much. So, what is phytochemicals? So, we know that what is medicinal plant? The medicinal plant containing phytochemicals. So now we have to see what is phytochemicals. The phytochemicals plant derived compound, and it should be less toxic with no side effect when we use it as a drug. So the phytochemicals is produced by either primary or secondary metabolites that include alkaloids, flavonoids, tannins, terpenoids, and cardiac glycosides, etc. These phytochemicals act as an antioxidant and also anticancers and the enzyme inhibitory effect, and also it protect the hepatocyte and the gastro and the renal productions and the cardioprotective effect. And it also act as an anti-inflammatory, and we use it as an antimicrobial activity. That's why we used turmeric for the skincare product. And also recent days, we used some Kabasura for the COVID, right? So it having these phytochemicals. So these phytochemicals are antiviral property. Some phytochemicals mimic like hormones. The hormones is produced by the body to regulate the physiological systems. So these sometimes for example, diabetes also inability of insulin, right? So we using some medicines for the control the sugar. So these medicines act as it's mimic like hormones like insulin. So it uptake of glucose by the alternative way by using the medicines, traditional medicines or phytochemicals. So what is polyphenols? Polyphenols is one of the phytochemicals. It produced by the plant as a secondary metabolites. It having polyphenols are it having the organic compound comprised both flavonoids and phenolic acids are ubiquitous in plants. So far, 8,000 polyphenols have been identified. They are short or about the 8,000 polyphenols. These polyphenols having their chemical structures are different colors from yellow to green or blue to red, especially so in fruit skin having this kind of listed colors, right? These are all the polyphenols. The major category of polyphenols is flavonoid, which is distributed as substance. The flavonol, just quercetin and camiphol are the most consumed flavonoids and are present high phenolic hydroxyl group are subclasses of polyphenols that include flavonols, flavonones, anthocyanidins, structures that give anticancer. Now we see the classifications of polyphenols. The polyphenols, they are classified into major two categories. One is phenolic acid and flavonoids and non-flavonoids. Phenolic acid include hydroxybenzoic acid like gallic acid, example gallic acid, and hydroxycinnamic acid, example ferulic acid. And non-flavonoids, stilbenins like resveratrol and lignans, hexylceranols and curcumins. Flavonoids include flavonol quercetin, flavonol apigenin, flavonol catechins, and flavonol naringenes, and isoflavonin, pyranine, and anthocyanidin like cyanidins. This is the classifications of flavonols and the structure of phenolic acid, including benzoic acid and cinnamic acid, derivative based on the C1 and C6 and C3 to C6 backbones. These phenolic acid present in the fruits and vegetables containing many free phenolic acid in grains and seeds, particularly in bran or hull. Phenolic acid are often in the bond form. Structure. This is the basic backbone of flavonoids, and flavonoid have C6 and C3 C6 general structural backbone, in which the two C6 units like ring A and ring B. Ring A and ring B are the phenolic natures. Due to the hydroxylation pattern and the variation in the chromin ring C, flavonoid can be further divided into different subgroups such as anthocyanin and flavon-3-olus and flavonones and flavonon and flavonols. Some flavonoids such as isoflavone and neoflavonoids whose ring B is connected at C3, C4 positions of ring C, like this. This is the structure of flavonoid moiety. Here, this R group is if R group containing H, we call it epigenin, and OH, if it is presenting on the R positions, it's luteolins. And the same flavonoids and flavonols, R group only decided which flavonoid it is: chemiferol, quercetin, mycetin, and isoramectidins. This is the procyanidins, the oligomeric procyanidins, like linked to the flavonoids, more than one. This is the some other polyphenol structures like resveratrol, curcumin, and rosmarinic acid, and gingeronol, and ellagic acid, so on. What is the source for the flavonoids and non-flavonoids? Non-flavonoids like ellagic acid present in strawberries, blueberries, and raspberries. Pomeranz like bell pepper, bok choy, and cereal grains, and broccoli. Flavonoids like anthocyanins, red fruits, catechins, tea and wines, and flavonoids, citrus, flavonoids, fruit and vegetables, flavonoids, fruits, vegetables, tea and wine, isoflavonoids presenting on the soya beans. So how much we are intaking of flavonoids? So how the people's are dietary intake of flavonoids? So it is not constant, and it may be vary according to the human races and country. So especially in Asian country, our people are taking more soyas, right? So the Asia countries in 10 to 35 gram per day, which is equivalent to the mean intake of 25 to 40 milligram isoflavone per day, with the maximum intake of 100 milligram per day in Asian country people. In United States, they are taking 1 milligram per day. The 1 gram per day, it consists of 16% flavonols and flavonoids and flavanols, 17% anthocyanins, and 20% catechins. Phenols intake in United States, Denmark, and Holland is about 20 to 25 milligram per day. And in Italy, they are consuming around 5 to 125 milligram per day. And in Finland, they are taking 28.3 milligram aspartame per day, like flavonoids. And also in Finland, their high amount of berries are eaten. It contains berries, and berries containing anthocyanin, right? Because the outer layer having this blue color. The blue color is due to the presence of anthocyanin. So that's why the Finland people are taking much more, like 82 milligram per day. And also Spain, they are consuming catechins and proanthocyanin dimers, and the trimers have been estimated around 18 to 31 milligram per day. In Germany, they are intaking of phenolic acid range from 6 to 987 milligram per day. This is the approximate rate how their people are taking the polyphenols in their dietary intake. Once they are intaken the phenols, how much it is absorbed by our intestinal systems, and how it is metabolized by our organs. Most phenols are probably too hydrophilic in nature. So that means it can able to easily penetrate the gut wall by the passive diffusions, but some membranes carry that could be involved in phenol absorptions have not been identified. The other way of systems like the unique active transport mechanism that has been described as a sodium dependent saturable transport mechanism involved in cinnamic and fluoric acid absorptions in rat jejunum. This is like previously reported how their phenolic substance or polyphenols are absorbed in the intestine. The mostly the polyphenols having glycosidic linkage, so that probably resist acid hydrolysis in the stomachs. So once it is at acid hydrolysis, the only A glycons and some glycosid can be absorbed in the small intestines. The maximum absorptions occurs 0.5 to 0.7 hours after intake of quercetin, 4 glycosid, and 6 to 9 hours after injections of the same quantity of routine, quercetin 3 beta retinoid. So this is the difference. So if the quercetin having the 4 glycosid means it can quickly absorbed by our intestine, but the same quercetin having the retinoid side, it take a much longer time to absorption. So the polyphenols doesn't mean that means the meaning of which it could be having the molecules. One report has demonstrated that the glycosid could be transported into enterocytes by the sodium dependent glucose transporters SGLT1 and hydrolyzed inside the cells by cytosolic beta glucosidase enzymes. So this is this is in observed metabolized and observed in the small intestine. The alternative pathway observed the polyphenols in the large intestines called lactase polymerization hydrolysis. This is like enzymes produced by the gut microbes, like bus border membrane of small intestine that catalyze the extracellular hydrolysis of some glucoside, which is followed by the diffusions of A glycans across the brush borders. Brush border means it's like a layer on the intestines. The polyphenols that are not absorbed in small intestine that reaches to the large intestine, there is microbial flora hydrolyze the glycosid and to the A glycans and extensively metabolize to the A glycans into the various aromatic acid in the large intestine. This is the structures. So how their microbes are metabolized, the polyphenols in the large intestines, and it's observed in the small intestines by this. This is like brush borders layer on the small intestines to observe metabolism and absorbs the polyphenols. So the excretions, once it is observed by the intestines and it transported to the liver to the metabolism, then it should be excreted through the urinary tract or from the bile tract. But the data are still very scary, even in animals. Rapid energy dependent transport system is active in Arctic endothelial cells for the uptake of mooring and a few other hydroxylated phenolic compounds. Metabolites of polyphenols may follow two pathways of excretion. One is through the biliary and another one is from urinary tract. Large and extensively conjugated metabolites, it could be, I think it could be a little bit more in size, are likely to be eliminated in bile, whereas small conjugated, such as monosulfate, are preferentially excreted in urines. This is the overall schematic diagram how their polyphenols are observed and transported and metabolized and excretions. Here we injected polyphenols and it absorbed both in small intestine and large intestine. Then enzymatic modifications observed polyphenols are enzymatically modified into the either methylation, sulfylation, and glucuronidase transferations and esterifications in small intestine and large intestines, microbial metabolisms like esterification, dehydroxylations and reductions, and decarboxylations. After these enzymatic modifications, it is transported to the portal vein. The portal vein directed to the liver, where the livers are best to metabolizing enzymes like methylation, sulfylation, and glycoronidase. After the metabolism in the liver, further it connected to the systemic circulations. The systemic circulations, it carry the polyphenol, metabolized polyphenol to the various organ or tissue where it is required, where the mode of mechanisms happening in by the polyphenols. Then it follow the biological effect. After then its role, it get back to the systemic circulations and transported to the kidney to the excretion. So whether this polyphenols having this side effect or toxic to the polyphenols, but in the first slide we have seen it's not toxic or less in toxic, right? The traditional medicine system, but still these polyphenols also having the toxic effect. Like if it is administered over, it may cause the carcinogenesis, genotoxicity, and thyroid toxicity, estrogenic activity of isoflavonoids, and anti-nutritional effect, interaction with the pharmaceuticals, drugs. Here we are having some studies showing chronic nephropathy was observed in rat with high doses of quercetin. If they are administered quercetin 2% or 4% with the food, they having this chronic nephropathic problem. Caffeic acid for another one studies, they are administered 2% in the diet, include 4 stomach and kidney tumors are developed in mice and rat. High intake of isoflavone have been associated with reduced fertility in animals with anti-luteinizing hormone effect among the pre-menopausal women. Proanthocyanin and elagitanin have been considered anti-nutritional compound, particularly in animal nutrition, because they have interact with the food and inhibit the several enzymes. This is previous literature shows the biological effect of polyphenols. So this is like a neuro, it having the good biological effect like neurodegenerative diseases. Like for example, curcumin, resveratrol, and catechins may protect against Alzheimer's disease, and ionic ion chelating effect of curcumin, mesitines having this having the good effect with the Parkinson's disease, Alzheimer's, and Huntington diseases also. Some flavonoids having the anti-inflammations effect like phenolic compound may prevent systemic or localized inflammations by restoring the redox balance to reduce oxidative stress and by modulating inflammatory response through the mitigations of cytokines pathway. So many research papers are showing phenols having the anti-cancer property. Even my thesis also the same. Aspirin and quercetin can able to prevent the formation of lung cancers. So here the flavonoid such as anthocyanin, catechin, flavonols, The isoflavonoids may neutralize free radicals and decrease cancer risk by arresting cellular growth in tumors. The specific type of cancer with evidence beneficial effect from polyphenols include colon, prostate, epithelial, and endothelial, and even breast cancer also having this good effect with using polyphenols, and the cardiovascular effect, and type 2 diabetes, especially for anthocyanin being the most sustained drugs to treat or control the type 2 diabetes. And obesity, the recent days obesity having the big problem in the society. So the polyphenol like catechin, resveratrol, curcumin are associated with the anti-obesogenic effect and potentially through adipocyte oxidations and inhibition of lipogenesis and the reduction in inflammations and increase in energy expenditures, leading to improved weight loss and maintenance. This is like a diagram of what I said in the previous slide. The polyphenols having anti-allergic, anti-inflammatory, anti-proliferative, antiviral, antioxidant, and pro-oxidant, and the mutagenic effect, and the carcinogenic, anti-carcinogenic effect of polyphenols. Actually, in 2002-2016, in the worldwide published or studied these polyphenols. Like here, you see the quercetin having studied around 21,605 research papers, and also resveratrol having the good polyphenols. Most of the people are studied, and the catechins and apigenin and luteolin having the good studies, good publications are produced during the 2000 to 2016. This is like the number of paper published in various countries like USA, much more in US researchers, and China, then Indian researchers are mostly focused on polyphenols. So, and also flavonoids like China much higher, US and India in the third place. And also the lignans, China, US, Japan are leading researchers to concentrate on lignans, and stilbens also concentrated US researchers. So this is a much more focusing area recent days about the polyphenols. This is like number of publications generated in last 100 years. From the 1940 to 2020, this is like increase in number of publications regarding polyphenols and the antioxidant in the past 100 years. The publication paper, those are registered in the Scopus databases in October 2021. Results retrieved from the query polyphenols and antioxidants. This is the commercialized polyphenols are available in the market, like quercetin capsule, tablet, and a gel in cream form also available. This is like the quercetin rich food component like berries and onions, apple. These are all the quercetin rich vegetables. This is the commercially available tablet, capsule, and gel and cream form of quercetins. This is one of the mechanisms how the quercetins are acting as an anti-cancer mechanisms. This is the signaling pathway, how they are regulating the regulated by the quercetin to control the cancer development by the signaling pathway inside the cells. The another one, polyphenols like chemiferol and the catechins and resveratrol rich source, and also the tablet is also available, protein tablet and capsules. About these mechanisms, resveratrol biological property or biological functions, it having the modulation of cell proliferations and induce the apoptosis. Its apoptosis means programmed cell death, and the modulation of angiogenesis and inhibition of metastasis and the modulation of redox status and suppression of adipogenesis and the stimulation of osteogenesis, modulation of mitochondria activity, modulation of DNA damage, modulation of xenobiotic metabolisms, modulation of glutamic metabolisms, and autophagy and osteogenesis stimulations. This is the overall possibilities, possible molecular targets by the polyphenols. Here the anti-obesity and diabetes, anti-inflammations, improved cardiovascular disorders, and improve gastrointestinal pain and anti-hypertensions, and also having the anti-cancer property. Actually, these are all the mode of mechanisms how they are controlling these enzymes or protein to inhibit by the polyphenols. Like, for example, antioxidant property, it having scavenging the ROS, RNS, and hydrogen peroxide and oxidants. This is the signaling mechanism, signaling proteins to inhibited by these polyphenols to control the progressions of cancer. So what is the challenge we faced when we used polyphenol as a drug? This is the basic challenge. First, we don't know how much dose we have to give, because it is having a good effect. If we administer overdoses with a long period, it also causes a carcinogenicity, what we see it in the previous slide. So for that, we have to know the best dose. So we don't know that we are in unclear how much dose we have to use. And also the possibility of side effect, we don't know. And how long we have to use this drug to get a toxicity, that we have to be clear. And intricate interactions with the pharmacological drug. So the people are asking whether they are taking with the allopathic drug, they are asking, why don't we take the alternative medicines, Siddha medicines, meanwhile the combinations with the allopathy? That we don't know. So we don't know how this allopathy drug and Siddha drugs are interacting and doing the best biological roles. That we don't know. We have to study it with these combinations also. And also poor clinical trials. So this is not easy to do in clinical trials in our country. Not only in our country, in the world wide, because the clinical trials, if you are tried with your drugs with human patients, we have a lot of formalities to clear. And also perfect molecular mechanism and the cellular signaling pathways, we have to study it more. And stability and low bioavailability. So how much we have to take and how long the drug binding to the proteins or enzymes. So for example, if you are taking drug in the morning and night, right? So, but during that 12 hours gap, the drugs are excreted by the urine. So that's why the recent days, the researchers are focusing irreversible binding of drugs to the proteins. They are focusing on that concept. So once you take a drug, it won't be easily metabolized. So it irreversibly bind to the proteins. So that means the minimum intake of medicines is highly focusing. That's why they say the instability and the low bioavailability. These are all the common challenges to use this polyphenol as a drug. This is like a simple diagram how we have to extraction and purifications of polyphenols. So you have to collect the samples and the extractions either by percolations or saxilate apparatus and separated by column chromatography and identified the compound by GCMS, ALC, UV or TLC or other method. The problem in the yield of phenolic from the selected source. So this is like how we have to improve to get a good quality of polyphenols, like apply appropriate genetic engineering techniques. So we have to use this cloning technique to focus the good quality of polyphenols, and also metabolic engineering and ribosome engineering and processing designing for improve the extractions of polyphenols. So about the conclusions, polyphenols, the most abundant class of substance with at least 10,000 different compounds, have become a focus of research over the past 16 years. All polyphenols having the common molecular mechanism of actions that able to inhibit cell proliferations, angiogenesis, down-regulation of endogenous antioxidants, and modulate transcription factor and their associated kinase. And finally, it prevents many signaling pathway to control the diseases. Clinical studies are required on the concentrations and bioavailability, efficacy of polyphenols, and the safety of these polyphenols, and we have to know the clear mechanism of actions of these polyphenols for treatment of for the therapeutic purposes. And also we have to know these absorptions, distributions, metabolism, excretions, and the bioavailability and biological activity of polyphenols in human body must be further examined through the research. Polyphenols, in particularly quercetin, rutin, tamifol, catechins, and resveratrols, are able to prevent the lipid overproductions. That means control the obesity and reduce the incidence of cancer, and normalize insulin sensitivity to prevent the type 2 diabetes, and decrease inflammation, tissue inflammations, and reduce the incidence of liver disorders. That means productive natures and infectious diseases also. Overall, polyphenols are the best target of research into secondary metabolites, although they are studied to pose the certain challenges. So thank you very much for this giving the wonderful opportunity. If you have any questions, please.
Thank you, sir.
Thank you so much.
Now we move on to the question session, sir.
Yes.
Can you tell what is the role of metals in extract preparation? See, while you are doing the preparation of your extract, you said that some metals have been added. What is the role of those metals in the extract preparation?
Not in the extract preparation, madam. During the treatment, they used metals they're using this mercury. Sometimes they used gold also, right? So they are combinationally used to treat the drug, but not in the extractions.
Okay, sir.
Extractions, they used the different solvent. So either our compound is sometimes it's not dissolved in water, right? So for that we used ethanol or methanol, acetones. So these are all the solvent we used to extract.
Okay, sir. Then what is the purpose of adding these metals after the preparation of the drug? What is the purpose followed by these metals?
Yes. Actually, the enzymes are required cofactor to activate. For example, magnesium's required to activate the enzymes. Sometimes manganese also, zinc also needed to activate the enzymes. For that, they used these metals combinationally to give the drugs.
Okay, sir. Thank you, sir.
Yeah,
One more
thank-
there. Sir, while we move for allopathy medicines, we can easily define the structure of the component present in the drug. But we always find difficulty in designing the structure while we move for preparation of the drug from plants or from other extracts. No, how can we solve this problem, sir? This is the major problem. We cannot protrude our Siddha or Ayurveda medicine to world. I think so that this will be the major
Once
problem.
again, madam, please. Sorry. Once again.
Sir, while we move for allopathy medicine, we know that crocin has this structural combination,
Yes.
and catazin has this structural combination. We can able to say like this, but when
Yes.
we move for our Siddha medicine, we cannot exactly give the structural composition.
No.
Can we able to give, sir? That is the reason why I think so. We cannot able to design a proper drug for treating this this COVID. That is why we have not been recognized. I think so, sir. What is your comment on this statement?
Yeah, actually, that's why, madam. Still, people are believing to using the plant medicines as a drug, but they don't know what is inside. Even we don't know what is inside. So we have to clearly we have to clearly isolate the drugs, and we have to treat with one by one. So which component is responsible for which kind of which kind of proteins to target. This is the very clear. We have to know. Still, you know the curcumin, right, from the turmeric. These drugs are precursor for the around 10 to 20% of all the drugs. So this is the precursor, raw materials to synthesize the drugs. I think you know that.
Yes, sir. I know that.
Yeah.
Another question is there,
But
sir.
we don't know, madam. We are not clearly know. We don't know. Nobody's know. So what is
Exact
inside?
structure
So
we
which one is
cannot. No.
Yeah, no. That's why we have to. That's why the researchers are focusing. So which components responsible for which proteins or which mode of mechanisms, that we have to very clear. Otherwise, until we couldn't focus, or we couldn't say this is the right mechanism of actions.
Okay, sir. Thank you, sir. One more question is there, sir.
Yes.
Sir, usually people say that alkaloids are highly poisonous. It will damage our metabolic activity, and sometimes it may also lead to death. That is the reason why, because some animals and even goat, they won't take some alkaloid plants. They will avoid some plants which is rich in alkaloids. How far this alkaloid is useful for our phytochemical active? It is highly active or act as a drug for us, for human being.
Yeah, like alkaloids, we have seen that it is, if you take the higher amount, definitely it could be toxins. It could be damage to your kidney or internal organs. So yeah, like depending on the concentration, madam. So that's why even the drugs also, even if we are using this paracetamol, right? If you take too much of paracetamol, definitely it could be poisons. For example, if you take a 500 milligram tablet 4, and you mix it with the feed and give it to the rat definitely will die in 1 or 2 days. That is highly poisonous. So the same thing, how much concentration you are taking, that is the matter.
Sir, if polyphenols, if we intake polyphenols for a long term, after how much time we can see its adverse effects, sir? How long it will proceed, and at what time duration we can see its adverse effects?
It's depending, madam. I'm not sure this much concentrations which leads to this much. So what they are given, like a 4 to 6% of quercetins to the rat. So I have said in this slide. So 4 to 6%. So if you are taking 10 gram of food means the 2 gram must contain this compound. For the 16 weeks, definitely it will give the effect. So the concentrations and the duration time. But if you are taking with these fruits, raw materials, that doesn't do anything. But if you are taking only the raw materials, I mean the purified product, that could causes the side effect. So they based on the concentrations and the time of period, ma'am.
Okay, sir. Sir, in Siddha and Ayurveda medicine, they give a time period of 30 days, 45 days. Is there any significance in those, in mentioning these days, sir? The days? For
Yes.
the drug, they are defining a particular day, 45 days, 35 days, 25 days, like that they are giving a time period. Is there any significance in that, sir?
Yes, ma'am. Actually, by the for example, if your blood cells lifetime is 120 days, right? So red blood cells lifetime is 120 days. So after 120 days, the cells will be die and generated new cells. The same thing, your body is regenerating all the cells in all the time. So once it's regenerated, it will take that much time to the entire systems. So that's why they are giving this particular period.
Okay, sir. Thank you very much, sir.
Thank you,
you
sir.
very much. Thank you, sir.
On behalf of management of Untitled Arts and Science College, I extend very hearty thanks to Dr. Kamaraj Sathu for elegant session and sharing with us his valuable content today. It is a very informative session. Thank you once again, sir.
Thank you so much, ma'am.